Excellent. Yeah.
So, hi everyone. Hi, Dr. Nabil. How are you doing?
Great. Thank you. Nice to see you. Excellent.
Same here. Same here. So, we'll be talking today about personalized medicine, right?
That's
So,
true.
be some sessions. Maybe we did not decide yet how many sessions they are, right? But at least it could be some of them. Really interesting for medical doctors, for biotechnology, for biologists, for anyone who is interested in this topic, to know
That's
how the
true.
medicine is evolving and how the progress of medicine and how is the future of medicine, right?
Excellent.
So,
Yeah. We are
yeah.
really proud to introduce that our sessions on the future medicine, and we will try to be as much as we can to make it for public engagement and also for professional. Yeah. Floor is
Absolutely.
yours.
Yeah. So let me introduce myself quickly. I mean, so I'm Lubna. I did my PhD in biology, molecular biology, and genetics. And after that, I did a master in bioinformatics. So I'm personally a computer scientist since some years ago. And I'm working now in the field of gene therapy or gene editing, mainly on the data.
Excellent. This is Nabil Haji. I'm a professor at University of Seville, Faculty of Medicine. After doing many postdocs and research fellow in different places, Karolinska Institute, Imperial College, Birmingham University, Queen Mary University. Our field of research is mainly cancer, trying to personalize medicine by clustering patients and identifying key genes, key pathways to be able to recommend specific and selective treatment for patients. Nice to meet you, Luna. Thank
Yeah,
you.
same here. Awesome. Nice to have you with me. So we can start with a very briefly introduction about how the personalized medicine evolved from probably from several years ago. We just am I sharing my slides, right?
Yeah, I can see them. Excellent.
Okay,
You
good.
just
Okay.
go to the next slide, probably. Yeah,
Yeah,
excellent.
yeah. Okay. So what is the personalized medicine and how we can precisely define it? So as you may know, each person is unique. So yeah, it has a unique genome, and basically when we say unique genome is unique behavior of this genome. So this genome is mainly has a lot to do with, so the change of the genome has a lot to do with environment, with everything which we call right now exposomic, means that's everything what we eat, nutrition, what we our sleep patterns and substances we eat. Everything is related to how the genome is reacting and how the expression of these of the genes and also the proteins and so on. So mainly when we say that, we need to give some definition to each person, right? So personalized medicine, that means it treats each individual separately. It treats this individual uniquely. So that's for we need to make some treatment for this person, unique for this person. So mainly what we are doing is tailoring this person or tailoring a treatment for this person based on the biology, basically, and the environment, as we say. Yeah. Do you have anything to
Exactly.
add?
Yeah, no, I think that's the right say to say. So yeah, definitely we are witnessing a medical revolution or evolution, if we can say, thanks to advanced, a lot of cutting edge technologies. You mentioned already genomics, but also we are in the era of artificial intelligence and the system biology. We are moving from the what we call the one size fits all. So we cannot give the same treatment based on the same symptoms, because the same symptoms can come from different diseases. So we cannot do the one size fits all approach. So we are entering in this era, what we call personalized medicine or precision medicine. You can call it what you want. So where we can prevent, we can diagnose, and we can provide better and precise treatment for each individual based on its molecular, settings. So this is not science fiction. This is a science fact, and it's changing lives already. So there are many rare diseases already saved based on this personalized medicine. Yeah.
Yeah, absolutely. Here we will just give an introduction how the personalized medicines evolved since already some years ago. So we might know already from this from the ancient times. They knew already about the blood, the yellow bile, the black bile, and the phlegm. And this everything was related to how the body was changing with the environment. And then later, in the 19th century, there is this emergence of modern pathology. And then it started to be interesting from the 50 and 60, between 50 and 60, when Watson and Crick they discovered the double helix of DNA. And then, of course, the new era of multi-omics started basically on beginning of 20. Probably you see the mouse, right?
Yes, I can see it.
Okay, yeah, awesome. So beginning of 2000, we had already some clinical application about the BRCA. This is the famous gene about breast cancer, and then it was also in 2005, I think, where Illumina started. So this is the first platform technology, which start with omics, and then we have basically the huge integration of multi-omics and clinical data from 2010 to 2020. And today, when we say today is about the AI, it's not really today. It started already some years ago. I mean, probably middle of 70s. But when we say today, because we have many data available, we accumulate actually. We have a history now of databases where we can use to train our data and to get a better outcome, and how we can use that better and better to make the personalized medicine more performant and more accurate, right?
That's a great slide because it summarizes the history of personalized medicine, what we call precision medicine. I think if we take two things here, how it was before, it's only based on intuitive medicine. So the practitioners are trained to identify common symptoms and prescribe, the same treatment for all. But then in the early 2000s, basically, we have what we call the science-based or evidence science-based medicine, where actually the clinical trials was introduced, what we called randomized controlled clinical trials. And that's actually the beginning of the personalized medicine, where we start identifying some patients' response and other not. That's what probably will develop in the following slides. Thank you.
Absolutely. So yeah, as you mentioned before, so how was that the trial and error to tailored therapy or to personalized therapy? Obviously, we can, as you say, Dr. Haji, we cannot say one treatment for everyone. So one treatment might, from the statistical point of view, it might be efficient probably for 50% or 60%. It does not mean it suit to everyone, right? So what happened, even if it is 10%, 20%, where we have a lot of side effect, where we have some new diseases, where we have new patterns, or the life is degrading after the treatment. So this start to be really not a treatment, but then a new diseases coming on. And this is basically where the personalized medicine comes to, not only to give this unique pattern for each one and give a treatment for each one, also to avoid these side effects, right? To avoid that's what we have here in the slide, these new diseases, and also, of course, we can get to know the diseases better. So more we know about the persons, when we know also about the diseases. So the treatment will not only be for these persons precisely, the treatment will be also some, let's say, data to train in later to have a better treatment for everyone. So Yeah.
That's really good point. I think for precision diagnosis, you need precise understanding of the disease to provide precise therapy. I think this is precise, the three precise here to reach the precise diagnosis and precise treatment is very important. And to reach that, you need to have a precise understanding of disease. That's why this era where they introduce science to the medicine, then they start to understand better the disease, and having those cutting edge technologies, you gather a lot of data that can allow you to understand better the disease, you better diagnose the disease, and then you provide better or precise therapies. I think that's the point here when we say, precision medicine, and this is basically the beginning of the precision medicine. Yeah,
absolutely, absolutely. So what is required actually? What do we need for these precision medicine or personalized medicine? Mainly for now, we need three main data. So the omics data, we'll go through them next slide. And we need the non-omics data, and we need the clinical data. So what is the omics, and what is this omics data? What is for? So mainly when you see the slide here, mainly
yes.
we can extract the tissue, we can extract the blood, and we can make a landscape or overview about what these how much data we can extract from here. So we can make, for example, some genomics. Genomics, it could give an output about mutations, variants. So these variant could be punctual variant, it could be structural variant, it could be, copy number variant. So they are kind of broad spectrum about variant. When we talk about variant, of course, duplication of chromosomes or deletion of chromosomes, and so on and so on. So then we have also the transcriptomic, when we say about how the genes are expressed, right? So this is we talk about mRNA. In many cases, we can also extract not only the mRNA or this messenger, it also can extract all the kind of RNA, right? This is the non-coding RNA and all these, again, this era of non-coding RNA, how the RNA is regulating other RNAs, so this not silencing, interference, etc., etc. Then we have, do you want to say something? Go.
No, that's fantastic. I think you summarized very well the omics here. I think there are some other omics, of course. We cannot cover all of them. We always talk about, probably you want to say metabolomics. I just stopped you there. Definitely, so to understand the biochemical, the microenvironment of tissues. If we talk about cancer, we always want to understand how the metabolism of the tumor is happening there. Always can indicate, some metabolites can indicate the stage of the tumor, the response to treatment, and the evolution of the tumor. But also the microbiome, identifying the key bacterial family, even a precise species could indicate the disease progress, but even the response to treatment. Something else what quite a few people miss is the immune system. So when we talk about single cell immune system infiltration into the tissue, if we have an example here, case study, or a case, or as example, tumor, the immune system play a major role actually in the treatment. So the no infiltration of key immune cells to the tumor could actually lead to resistance to treatment. And also the polarity of immune system, really important if it's pro-tumoral, anti-tumoral. So all of those layers of data, they are really important. And I think the beauty is actually to integrate all of them to understand better the tumor. But I think there's more big data that we are talking about, not only omics data. I think probably you want to share with us another slide here.
Absolutely, absolutely. There is non-omic data, but I think also most of the persons are aware about non-omic data, right? So we're
Yeah,
talking about
true,
lifestyle,
true.
how the persons are living, how the nutrition, the movement. I mean, sport and being movement, being how is your lifestyle actually? How is it your daily life? Your sleep pattern? Do you have stress? How do you sleep? Do you sleep regularly? Do you sleep early? Late. So all these kind of habits and also the substances used like drug, alcohol, and so on, all these kind of non-omics data are very, very important to make again these not the personalized medicine. Uh, uh, um,
I
I
think
mean,
that's fantastic. You mentioned the omics and non-omics. We need to highlight the point here. I think we have a long discussion together for many times now about the modifiable risk factors if we focus on cancer and the non-modifiable risk factors. I think when we talk about non-omics data, we are talking about modifiable risk factors. Of course, you could modify the lifestyle. You can protect yourself from infections, you can sleep well, you can eat well, all those you can modify them. However, when we talk about genomics, the mutations you cannot modify. So the disease here, if we talk about cancer, is a combination between modifiable risk factor and non-modifiable risk factor. And then the understanding of these two risk factors, the two types of risk factor, will clarify our understanding of the disease, in this case, cancer. Yeah, that's fantastic. Thank you.
absolutely, absolutely, absolutely. I mean, the inheritance parameter is very important as well.
Yeah.
Then we have the clinical data. So the clinical data, we can talk about everything related to age, to sex, to where this person is living. Is he living in which area? Geography, for example, is also
True.
another important factor. Then we have these everything related to imaging and radiology, the history of the person itself, which kind of substances she was taking, drugs. I mean, when it takes drug, I mean medicinal drugs, or a kind of diseases she had before, or surgery. So everything related to the history of this person. The allergy, of course, this is also something really important to add. Anything to add beside
Yeah, I
that?
think I think yeah, I think now you mentioned the omics data, non-omic data, and clinic data. I think the beauty here, how we can integrate all those layers to be able to stratify the patients basically to put them in subgroups, and basically you can predict, the survival, which is mainly the prognosis, the type of the treatment, and even the response to treatment. So I think the integration of all those large data will be really useful to personalize the treatment, or to personalize medicine. So that's the beauty. Before it was intuitive, now basically we're based on molecular data and also clinical data, and everything together will help us to stratify better those patients, because we always miss a small group of patients. They are not responding, as you mentioned already. Maybe they are not responding, or they show some toxicity. Sometimes basically they don't really respond. So by integrating all those data, we can really help be
Indeed.
much more precise. Yeah.
Absolutely. Um, So we would like to go a bit into the personalized medicine in the field of tumor, since you are researcher in these areas, or you
Yeah.
are the better person to tell us a bit how can we use this personalized medicine concerning cancer.
Yeah, we understand personalizing medicine can be in different type of diseases. Our area is mainly cancer. Well, we use an in silico tool as the first tool, having two types of data: the public domain data. We are talking about large data, thousands of data based on the type of cancer, but also we use the clinical trial data and the laboratory data. So first, uh, is actually how to search for key features to be able to map the tumor and to provide an ID tumor. So to map the tumor, first you need to identify like key hub genes, what we call the key genes.
Like
and
P53,
pathways.
for instance. Like
Sorry,
P53, for instance, this is the
that's
God gene,
true.
as we say.
Yeah, I mean, the 50% of tumors, they are mutated p53, and p53 is tumor suppressor gene, and play major role. It's what we call it like a Swiss Army knife. So it can do a lot of things. So if it's mutated, could collapse many biological processes. So if you identify this gene, if it's mutated, you understand very well actually what is the consequences. But of course, for us, we are dealing with, thousands of data. What we do basically comparing the healthy tissue, relevant healthy tissue, to the cancer tissue, and identify the alteration of gene expression. Some of the genes, they are upregulated, other downregulated, and even sometimes they are silenced. And then basically what you do is to identify the key features of those up and down regulated, which basically allowed you to cluster those patients based on the alterated pathways. And then and this way you could basically recommend the treatment, and then you can even follow the progress of the tumor. We are not only interested in drug resistant features, but also the features that can decide the survival of the patients. So that could help us also to provide better treatment and extend the life of those patients. Also identifying some key features that can help us to detect early the disease, to subtype better the disease, because we know the major problem in some type of cancer like breast cancer, the triple negative, we still we don't know how to subtype those. But if you use all those omics and non-omics data, you may get better stratification, and you can reach better treatment to large subtypes. So there is a lot of positive things from this classification of the hub genes. So for us is to identify key genes that we can use to recommend for treatment, but also we can make the survival better prognosis. So that's basically what we can say.
Yeah, so mainly it's using the genes, or the output of the genes related to the all kind of pathway, basically fetching in the database where these kind of gene product is involved. So, and then you can cluster. Probably clustering is the most appropriate, because we know that sometimes one gene could be involved in different pathways. So in this case, classification would be a bit narrow, but if we use clustering, that we can give a bit better flexibility, probably.
That's true. I think what we do basically, when you do the expression profiling, as I said, comparing tumor tissue to healthy tissue, you find some upregulated genes and other downregulated. We're interested in the most interconnected genes, the one that if you inhibit or you activate, depending on the context, you could restore the pathway. And those hub genes or pathways, they are the one who will help us to cluster those patients. So when the patient come to us, basically what we do, we just position that patient in specific cluster. But first we need to provide an ID tumor of that patient, and we will know exactly what is wrong with the patient, and we just put the patient in the right cluster to be recommended specific treatment, either drugs or diet or other things, which can restore that pathway. Yeah.
Wonderful. Yeah. As you say, probably we talked already about that. Yeah.
Yeah, I think here what we can add.
Yeah.
Well, what we can add, basically all the data that we gather, either you can do it supervised, because as a biologist, you understand the pathways, you just be guided with the data, but also we use artificial intelligence in non-supervised way to identify hidden features that could be really interesting for small number of people in a big population. So that's what we want to say here.
Absolutely. I think the deep learning here will be a key, actually, because since you are handling a lot of data and this data is not that clear, so the only thing, or the only way we can solve this problem is the AI. I mean, using the deep learning, training the data, and taking again, and more you have, more data you have, and more better accuracy and precision you have, right? So this is basically another dilemma in medicine. This is this specificity and sensitivity and this accuracy and precision, where we're always, when we have a treatment, how accurate is it? And so it's still will be still a struggle, and this is a long way actually to resolve this. Okay, now how can we use that, for instance, in the future? So basically, it would be we will have something like dictionary, probably starting from embryology, right? So the fetus is now, we're not talking about even the fetus. I don't see the baby. I'm looking even before that, and the womb. So how the fetus is and how the baby is, and taking each sample and extracting all these kind of data we're saying, because we don't have to forget that transcriptomic is changing, right? Proteomic is changing. Probably the genetic itself is somehow much more stable than transcriptomic and metabolomics, and so everything should be taken in steps that we can collect this data. And after that, the person is probably once she's 30, 40, 80, whatever, she has a dictionary. So now the question is how the doctor can use that. So is it
This is a big challenge. So I used to teach this in Queen University. You know, the challenge that will face the future doctors, because they will be faced with millions of publications, millions of data. So how to extract the right data at the right time to provide the right treatment. So we need to teach those students basically new skills. We don't want them to memorize things. Of course, memorizing is always good, but at a certain level, when you're dealing with millions of data, you cannot memorize all of this. You need the tools. You need AI tools to extract the right information at the right time. So they need to learn, all these AI tools, and to be able also to integrate omics data and non-omics data, of course, with clinical data, to be able to, as you said here, the 4P, which is fantastic. The future medicine is predictive, preventive, personalized, and, participatory. So you need to be, involved, 100% with the patients. And as you said, the future is to create a story of the patient from the beginning to the end. I think we are talking about exposome and all of this. I think this is a fascinating, but we cannot really go to the details. Okay, we are over all the time.
And probably even if we go into a lot of details, there are many things which there will be in the future we cannot predict, obviously. True. But now that this is fantastic. This is something which is revolutionary, which started already. It's not, as we said before, it's not science fiction. It started already, but it is continually evolving and developing. And as you may know, the more data you have and better training you have. Exactly. And of course, we need computer scientists, we need engineers, we need biologists, we need experts for that, but all of them make a team to make this personalized medicine possible. Excellent. So I think, Dr. Haji, we will conclude with that, right?
Yeah,
so
you can go if you want. You have a
I think it's okay for now. Probably this is, as you say, this is the first session. We can make more sessions. But this is just to give an overview. If you want to add anything.
Yeah, I think for the conclusion, personalized medicine is not just scientific breakthrough. I think it's more responsibility, but I think it's a evolution of science and revolution at the same time. If we can predict how we will benefit from treatment for each individual, also we can minimize the risk we can also increase the benefits, and then for every individual. Well, thank you very
Absolutely.
much. Yeah.
Absolutely right. Okay, so with that, we finish our session for today, and see you next time. Thank you so lot.
Excellent. Thank you.
Okay.
Bye-bye.
Bye-bye.
Bye.